Welcome to our channel Mootechie. This is our visual-heavy biology series designed with a minimalistic, modern, and exam-oriented approach. Now, let's begin our comparative biology series focusing on prokaryotes versus eukaryotes. This is a comprehensive visual guide to understanding the fundamental differences in cellular structure, organization, and function. On one side, we have prokaryotes, which means before nucleus. This group includes bacteria and archaea. They are known for their simple structure and are exclusively unicellular. On the other side, we have eukaryotes or true nucleus. This group encompasses plants, animals, and fungi. Their cells are characterized by complex internal compartments and they can be either unicellular or multicellular. In this series, we will break down the key comparison points: cell size and organization, the nucleus and DNA structure, organelles and complexity, cell wall composition, and finally, reproduction methods. Let's dive into our first comparison point: cell size and organization. First, looking at prokaryotes. They are small, typically ranging from 0.1 to 5.0 micrometers in size. Their small size is linked to their simplicity. They are mostly unicellular and have a simple organization with no membrane-bound organelles inside. Their internal machinery is basic, consisting of rudimentary or simple proteins. Their ribosomes, which are the sites for protein synthesis, are the 70S type. These are smaller and float freely in the cytoplasm. Now, contrasting that with eukaryotes. Eukaryotic cells are significantly larger, typically between 10 and 100 micrometers. This larger size allows for greater complexity. They can exist as single cells or form multicellular organisms with specialized tissues. Inside, they have extensive internal compartments called organelles. The cytoskeleton, which provides structure and enables movement, is made of complex microtubules and filaments. Their ribosomes are the larger 80S type, found in the cytosol, and they also have 70S ribosomes inside some of their organelles. Moving on to the core of the cell, the nucleus and DNA. This is perhaps the most defining difference. For prokaryotes, a true nucleus is absent. Instead, their DNA is located in a region called the nucleoid. Their genetic material is typically a single, circular, and naked DNA molecule, meaning it isn't wrapped tightly with many histone proteins. They usually have one main chromosome, making them haploid. Extrachromosomal DNA like plasmids is very common. Their gene organization often involves operons and they have very few introns, which are non-coding sequences. For eukaryotes, they possess a true nucleus surrounded by a protective nuclear envelope. Their DNA is organized into multiple linear chromosomes that are densely packed with histone proteins. Organisms are often diploid or have even more sets of chromosomes. Extrachromosomal DNA is rare. Their gene regulation is more complex and introns are very common within their genes, requiring sophisticated processing.
Now, let's explore the world of organelles. This is where the internal complexity becomes very clear. For prokaryotes, membrane-bound organelles like mitochondria are absent. Any metabolic processes happen via membrane enfoldings or enzymes in the cytoplasm. They only have 70S ribosomes. For movement, they use a simple rotary flagellum made of a protein called flagellin. Storage is handled by simple inclusions like granules. For eukaryotes, a variety of membrane-bound organelles are present, including mitochondria, the endoplasmic reticulum, and the Golgi apparatus. Plants have the additional organelle, chloroplasts. They contain both 80S ribosomes in the cytosol and 70S ribosomes inside mitochondria and chloroplasts. They have complex whip-like structures for movement, called cilia and flagella, which have a sophisticated 9+2 array of microtubules. They also use various vesicles, lysosomes for digestion, and peroxisomes for detoxification. The remaining values in the table follow this pattern of contrast and can be referred to directly.
Next, we'll compare reproduction and growth methods. Prokaryotes reproduce asexually only, primarily through a process called binary fission. This allows for very rapid division, sometimes in just minutes. They can also share genetic material through horizontal gene transfer methods like conjugation. Their process is simple and doesn't require a mitotic spindle. Some can form durable endospores, but these are for survival in harsh conditions, not for reproduction. Eukaryotes, in contrast, can reproduce both asexually through mitosis and sexually through meiosis. Their cell division is generally slower, taking hours or days. Genetic variation primarily comes from sexual recombination during meiosis. The process is complex, utilizing a spindle apparatus to separate chromosomes. They also produce reproductive spores, like in fungi, and specialized sex cells called gametes. Our final comparison point is the cell wall. In prokaryotes, most species have a cell wall. In bacteria, this wall is primarily made of a unique molecule called peptidoglycan. The thickness of this layer is crucial and is the basis for the gram-positive and gram-negative staining classification. Many also have additional protective layers like capsules. In eukaryotes, the presence of a cell wall varies. Animals lack a cell wall entirely. Plants have a cell wall made of cellulose, while fungi have one made of chitin. Instead of a wall, animal cells have an extracellular matrix or ECM for support. Plants may have a waxy cuticle. Additionally, eukaryotic cell membranes often contain sterols like cholesterol, which provide stability. Thank you for watching. We hope this visual guide helped clarify the key distinctions between prokaryotic and eukaryotic cells. For more exam-ready content, please subscribe to Mootechie and follow us on social media. Keep exploring and best of luck with your studies.